So in the last class, we have learned about the chemical nature of genetic material. And in this class, we will learn the chemistry of it. So basically, we are learning about the chemistry of nucleic acids, which you already have studied, but we will have a quick revision on that. And then we'll discuss about Chargaff's rules, and then hopefully we'll finish Watson and Crick model as well. So while you are going through this lesson, and also I'm hoping that you're reading the textbook as well, it's very important that you get in sync with your textbook so that your understanding will be much better and you'll be able to follow through the course well enough. So you are supposed to know the chemical structures, usually up to 5 marks or 5 to 10 marks. Could be dependent on your ability to memorize and reproduce chemical structures along with numbering. Okay. And you should be able to describe the structure of DNA, the double-stranded, and so on. Upon that, you should be able to calculate the nitrogenous basis composition and so on, which we will practice provided we finish enough today. And so there are going to be a lot of things that you want to have. These two will be on basis of recalling, and your ability to calculate will be a little bit higher. So I would want you to practice, study the chapter well enough. So I think we all know that nucleic acids contain a nitrogenous base and a sugar, a base, a sugar, and a phosphate. Right? These are the key 3 things that are present in nucleic acids. So nitrogenous bases can be divided into purines and pyrimidines. And in that, you have 2 different types of purines: adenine, which is represented by capital A, and guanine, which is represented by capital G. Pyrimidines, you have cytosine, C, and thymine, T. If you were describing about RNA, instead of thymine, you would have had uracil, right? That's in the RNA. The rest of in RNA, the difference between RNA and DNA in terms of nitrogenous bases is only about thymine and uracil. So the chemical structures, we will have a quick look, and I want you to practice several times so that you can reproduce those figures or the chemical structures in your text, in your answer scripts. So purines are, they have 2 rings. One is a 6 member, and the other one is 5, right? You have, anytime you represent like this, that means it is a C, and here is another C, C, and C, and so on. And then if it is represented by N, there is a nitrogen atom in those places. One has to remember the purine structure. This is usually the purine structure. And if you have, say, the numbering also happens, numbering starts at this N. 1, 2, 3, 4, 5, 6, and then go 7, 8, 9. That is how you have to remember the position. So N6 purine, N6 purine amino, okay? N6 amino purine, that means this is the 6th position, and if I have an amino group, that is this one, NH2, then it is adenine. Okay, so you can always, I think I would encourage you to take your books now, have a pen with you, and try to solve it. Try to memorize it your own ways. Different people have it different ways, so you can do it your own way. 1, 2, 3, 4. That is how I remember. I'll put N and N here. and then you have N and N. So you can always solve for the double bonds. And this is the sixth position, 1, 2, 3, 4, 5, and 6. If amino group here, then it is adenine. Okay, I did not solve for the double bonds. You can always do those. You should always do that. Then instead of if you knew this structure, the purine structure, you can derive adenine from it. And once that is known, you can always, so the adenine that is present on sixth carbon, you can just say 1, 2. In guanine, what is the difference? You have amino group and a keto group here. So I can quickly again draw the same N, N. And instead of amino group, I would have drawn a keto group, and here I would put amino group, and that is guanine. This one would have been adenine. Again, double bonds, you can always solve for that. So you have to practice these things. There is no other way or logic to explain you everything. You have to remember that there is a 6-member ring, and then there is 5-member ring. That is the first thing to remember. Second thing to remember is, where do you put N and N? There is position, okay, N here, and there are 2 more N's. There are 4 nitrogen atoms in this. And numbering always starts at this N here. Okay. So similarly, you can also you have to remember for pyrimidine as well. Pyrimidine, you have cytosine and thymine. I like to think of uracil. That's much easier for me. So I will try to do that and then see how far we get. Uracil is present only in RNA or not in DNA. So all the pyrimidines have only one circular or ring structure. In purines, you have 2 ring structures, right? Here and here, 2 ring structures. Whereas in pyrimidines, you have a single ring structure. It is a 6-member ring structure, and it has 2 nitrogen atoms. The numbering happens in the reverse order: 1, 2, 3, 4, 5, 6. Again, if you are referring to multiple books on just the chemistry of it, in some cases they would have put this in inverted position, and then the numbering could be different. What I would suggest is, because you're supposed to memorize, follow one textbook for chemistry of nucleic acids, follow one and keep only that. Don't confuse yourselves by referring to multiple books on that. If you have followed the other one and not Watson or whatever I am showing, I am fine with that. You can do whatever you're already following. Ensure that the placement of the nitrogen atoms and the numbering is correct. So I always remember it. I remember uracil, okay, because it is simple. You just have to put 2 of this like this. Again, I didn't solve for the double bonds. You can do that. Okay, so the numbering starts 1, 2, 3, 4, 5, and 6. That's uracil. Now, this is, I think you can, if I have replaced this with cytosine, with an amino group, what do I get? I will get N. I am putting the keto here. On this position, at position number 4, I am putting an amino group instead of an oxygen atom. So what am I getting now? That is nothing but cytosine, what you see here. If I do this is uracil, right? 2 oxygen keto groups here, something like this. If I add a methyl group here, then it is thymine. So I have my own ways of memorizing things, and you can have your own ways. And the end, you have to ensure that you are able to depict these structures correctly placement of nitrogens, and the numbering should, and the groups and numbering should be proper. Okay. If you like this, you can follow, or else you can come up with your own base. So that is about nitrogenous bases. Purine. Purines, there are two purines, adenine and guanine, right? 6-aminopurine is adenine. If you replace the amino group in the to the second position, and in the sixth position you add a keto group, that is guanine. So if you want to memorize, you can memorize adenine, because from there you can always derive guanine. You don't have to memorize each one. So you have to memorize one structure. From there you should be able to derive the rest of the things. For pyrimidines, I remember uracil, which is present in RNA or not in DNA. The difference between uracil and cytosine is it has a amino group and the fourth position, this one. Then it is cytosine. If I take uracil as such and methylate on the fifth position, sorry, say something like this, or here that I have shown, if I have add a methyl group at that position, then it becomes thymine. So for memorizing, if you find it helpful, I would suggest you to by heart adenine, structure of adenine and structure of uracil. But you should also know how to derive the rest of the bases. Okay. When I said there are groups like amino group and keto group, I'm not going to emphasize on it right now because we will do it at a later time. But I want you to anyway introduce something here. They are called as tautomers. This is an amino group, NH2. And sometimes it may become deprotonated, or one hydrogen atom may be lost, and now you have NH with a double bond to this carbon. And that's called as iminoform. These, whether they can donate or accept electrons, is the key to some of the things that we will discuss in a later time. But right now, you should understand that the amino group can also exist in imino group, okay, occasionally. And the keto group that we described like this, oxygen, that can exist in enol form, that is OH. This is the OH. Okay, during different physiological conditions or metabolic or reactive oxygen species or change in pH, many things could alter this. But predominantly what you see is in DNA, the nitrogenous bases, you will see amino group and keto group. But sometimes the amino group might become imino group. Those are called as tautomers. And keto might become an enol. And they are involved in generation of mutations that we will discuss sometime later. So now we have learned about nitrogenous bases. We have discussed, now we have to develop further more complexity bases. Then we know that it has sugar, and the sugar is ribose or deoxyribose. In DNA, we have deoxyribose. Okay. And there is also phosphate. How do you, for me, the way to memorize ribose is to draw a 5-membered link like this. This one anybody can write just like knot form, right? And then you put HOH and so on. And here CH2OH. So it has 5. Ribose is a pentose. You have carbon 1 here, 2, 3, and 4. And 5. This is the 5th carbon, CH2OH. So you and then the sugar plus the nitrogenous base plus, I'll just write base plus sugar will give you nucleoside. Okay, and nucleoside plus a phosphate group will give you a nucleotide. Okay, so remember this. You can say, I will try to write again, a base, nitrogenous base, and you add a sugar, that is ribose. I'll just put as ribose. Ribose is present in RNA. What is the full form of RNA? Ribonucleic acid. And DNA is deoxyribose, deoxyribonucleic acid. So base plus ribose, you will get a nucleoside. Nucleoside, if there is a phosphate added to it, then you will get a nucleotide. So there is no escape but to learn these terms properly. You have already learned this, but anyway, I'm going through because you should become become familiar with the terminology. Because without terminology, we will not be able to follow the textbook properly or even the class. So right now, when I said base plus ribose or deoxyribose, yeah, I need to differentiate between ribose and deoxyribose because I was trying to explain the structures. So CH2OH, and you have OH, H, OH, OH. So the carbon numbers, the when you are discussing about the numbering of ribose or deoxyribose, you always see one like this they put, right? One prime, you call it as prime. I don't think calling it as one dash is right or two dash. It is two prime. Don't call it two dash. So you have to write, why do we add this prime or this one? That's a headache. But we do it because we want to distinguish between the numbers that are numbering of the sugars from that of the bases. If you want to recollect the numbering of bases, we are putting 1, 2, 3, 4, 5, 6, 7, 8, and 9. So if I said 2 carbon, you of 2 carbon of adenosine. Adenosine is a nucleoside. That means it has adenine plus, say, deoxyribose. If I said 2 carbon of adenosine, then I am meaning this carbon. I am not meaning this carbon. So the distinguishing, if I said 2 prime of adenosine, then it would mean that we are talking about the carbon, second position carbon that is present on the deoxyribose in this case. So the prime here is to denote the numbering of the sugar or the deoxyribose or ribose, as the case may be, to distinguish from the numbering of the bases. Okay, I hope you will be able to remember that. Now, this is the structure that I have written here is ribose. Ribose is a pentose. And what you have here is 1 prime, I have to call it, 2 prime, 3 prime, 4 prime, and this is the 5 prime. So deoxyribose is, that means in this case, the 2 prime O is removed. What you have here is just H. That's why we call it as deoxy. We are removing one oxygen atom, and we are removing that oxygen atom from the 2 prime carbon, or the 2 prime, the OH attached to the 2 prime carbon. I hope you will be able to remember that. So quickly, if you want to practice ribose and deoxyribose, it's simple. You do it. It's almost like you are writing a Rangoli. 1 prime, 2 prime, 3 prime, 4 prime, and this is 5 prime. That is ribose. So ribose has OH. Right? You have to remember that this 3 prime OH and 2 prime OH are very critical, are crucial in all of our molecular biology. More so the case with 3' OH. Remember that when we are learning about replication and so on, it will come so many times. So I want you to understand the chemistry and try to by heart these structures, so you'll be able to reproduce these pictures better, and your understanding will be good. So here is the deoxyribose, CH2OH. Everything is same. I'm not changing anything. Only difference is I'm putting H here. Deoxy, one oxygen atom is removed, and that is at the 2' carbon. That is deoxyribose. So basically, what is happening? The glycosidic bond, the bond that is formed between, I will erase this. The bond that is formed between the nitrogenous base and the sugar is called. This is the bond that is called as glycosidic bond. I hope you remember this is a carbohydrate. It is forming a bond, so you call it as glycosidic bond. And you are just removing the OH from the 1' carbon and the hydrogen atom. You are removing a water molecule, H2O. You have 2H and 1 O. Similarly, so by doing so, you are forming a nucleoside. What is a nucleoside again? Nucleoside is base plus ribose or deoxyribose. I'll call it as deoxyribose. Okay, and the next water molecule is removed from the 5th carbon, the hydroxyl group that is present on the 5' carbon of deoxyribose, and 1 H from the phosphate. Phosphate has 5 bonds, so double bond here, 1, 1, 1. So this is phosphoric acid or phosphate bond. I didn't tell, so I'll first put 5 bonds and then just solve it for this. If there is typically oxygen will have 2 bonds, so you can put, but often they would deprotonated and be in negative charge like that. Okay, so once you have added the phosphate group is added on the 5' carbon of deoxyribose. The glycosidic bond is formed on the first 1' carbon, right? This is the glycosidic bond. So what is a nucleotide? Nucleotide is a nucleoside plus a phosphate group. Okay, now we have the monomers that are required to form DNA or RNA, the polymer. DNA and RNA are polymers of these subunits. What are they? I think we will see it in the next slide. But yeah, I will just give it here. So this is DAMP. What is the difference between DAMP and AMP? Adenosine monophosphate, M. Because there is only one phosphate group added here, so you call it as monophosphate. I am not saying adenine, I am saying adenosine. That means adenosine includes adenine bound with a ribose or deoxyribose. If I put a D here, that means it is deoxyribose. That will be DNA. Whereas if I did not put D, that means it is ribose. So adenine plus ribose will give you adenosine. Adenosine plus monophosphate, that is AMP. DAMP would indicate deoxyadenosine monophosphate. Okay, these are subunits of DNA, whereas AMP would be a subunit of RNA. So here we have discussed just about adenine because we took only adenine as a base, but you can also have CMP, TMP, or I have written something wrong here. What is wrong here? Can you tell me this? Anybody? I have written wrong in this box in the rectangle here. Can anybody tell me what is wrong here? No? So as I told you a little while ago, thymine is always present in DNA, and uracil is present in RNA. So pyridine monophosphate is fine, but thymine should have been deoxy, isn't it? Not that this compound would not form, but in the light of nucleic acids that we are actually studying, thymine is present only in DNA, so it usually is associated with a deoxyribose and not a ribose. So I hope you'll be able to remember those. So ATP, AMP, GMP, CMP, and UMP will be in RNA. If you have to talk, these all can be just written as NTPs, or I'll write as NMPs, nucleoside monophosphates. Collectively, they can be called that way. DAMP, DGMP, CMP, and DTMP. These would be found in DNA. And then you can call it as DN, deoxynucleoside monophosphates. Okay, so what is the difference between RNA and DNA? You see, what is the difference between ribose and deoxyribose? One oxygen atom, right? You have one oxygen atom difference between ribose and deoxyribose. What is the difference between uracil? I'll go back so that you will be able to recollect here. What is the difference between cytosine and uracil? All the difference is one methyl group. Chemically speaking, if you look at, if you have DNA and RNA, then the difference is just one oxygen atom in the 2' carbon of ribose, and you have one methyl group in the 5', you should not say 5', I should say 5th carbon of thymine. That is the only difference. We'll recollect it if you have confusion in that. So I hope you understood this, and I would want you to remember some of these bases. You don't have to mug it up, but I would like to remember the molecular weight of 331 daltons. What is a dalton? Anybody? Sorry. What is a dalton? One-twelfth of carbon, or you can just say one weight of one hydrogen atom. Okay. So there are also here, I just described as a monophosphate. I represented DNMP, deoxynucleoside monophosphates. There can also be many others. You can have DNDP. DP is diphosphate. And similarly, you can have DNTP. That is deoxynucleotide triphosphate. So it all depends on the how many phosphate groups are added to the 5th carbon of deoxyribose. This is the 2nd carbon, 2' carbon of deoxyribose. There is no OH. There is only H here, no oxygen atom. So phosphate group, one phosphate group, then it is monophosphate. Two phosphate groups, then it is diphosphate. Three phosphate groups, then it is triphosphate. There is a nomenclature for this as well. The first the carbon directly attached to the ribose is referred to as the alpha phosphate. The second one added is beta phosphate, and the third one is gamma phosphate. So for a quick review, I would want to go through this. I hope you realize that the adenine that is given here, it is in reverse order to what I have shown. That is the confusion I was telling you about. So now if I have to number, should start 1, 2, 3, 4, 5, 6. 6 aminopurine is adenine. Okay, 1, 2, 3, 4, 5, 6, 7, 8, and 9. Okay, this is the glycosidic bond. And here you have 1' carbon of deoxyribose. This is 2', 3', 4'. This will be 5'. And then you have alpha phosphate, beta phosphate, and gamma phosphate. Okay. Those are some of the things that you are supposed to learn and memorize them. I think the simplest way to do it is practice it for a week, or maybe 4 times a week, and occasionally once in a month or so, I would want you to look them up. That is, recollect, revisit the structures so you can memorize them much more for a longer time. What is a trinucleotide? Until now, we described about single one base, one sugar, one phosphate, or 3 phosphates. When you say trinucleotide, one nucleotide, if you say, it should contain one base, one sugar, and one phosphate, right? That is one nucleotide. And if you have 3 of those, then you call them as trinucleotide. So I will try to draw. This is one nucleotide, what you're seeing here. And this is another nucleotide. This is second one, 1, 2, and the last third one. There are 3 nucleotides. So and they are covalently linked. So you call them as a trinucleotide. All of them will have. Can anybody? Yeah, it is a DNA because you have thymine. It is DNA because there is no OH on the 3' carbon. Sorry, 2' carbon. This is 2'. This one. Okay, and I hope you remember 5-methyl uracil is thymine. So if you just add a methyl group to uracil, that is thymine. Okay, this is adenine. Here you have cytosine. So why this is important is DNA or all nucleic acids have a polarity. Polarity in the sense it is what is the end of it. So this is one nucleotide, right? You have these I have distributed. So 1, 2, and this is the third part. They are in a sequence. Should I read this sequence A, C, T, or should I read it T, C, A? So what you have to remember, these things will become very crucial for your understanding or following the subject itself, is there is one end of this single strand DNA is having a free 5' phosphate. Why do you call it 5'? Because this is the 5th carbon on this deoxyribose. This would have been 4, 3, 2, 1. I have to always put this prime, right? One end of the nucleic acid here that is shown has a free 5' phosphate group. That is, you're saying that there is a phosphate group at the end of this one, this carbon. And the other carbon is having a 3' OH. This would have been 1', 2', 3', 4', and 5'. These are the free groups that are reactive. So phosphate is electronegative, and OH can become also positive or donate a hydrogen atom. So basically, these are the 2 ends all nucleic acids are represented usually just by a line. And the key things that they put is always you have to write what is 5 prime, and the opposite of it, opposite end would be 3 prime end. You should all, you should, if you are talking about a single strand DNA, this is a single strand DNA that you're seeing here. This one is a single strand DNA. You should always start with 5 prime end. You should not represent it as, that will be wrong representation. Not that you shouldn't do, but it is not followed. Suppose if you don't represent 5 prime and 3 prime, and you just draw a line, it is generally assumed that it is, this is the 5 prime and this side is the 3 prime. But don't leave anything to ambiguity. Always indicate the 5 prime and 3 prime ends of all DNA or RNA molecules that you are representing as a part of your any depictions or diagrams. So remember that all nucleic acids have a polarity. Polarity means you can tell the difference between one end and the other end. And always we should represent it from 5 prime end to 3 prime end. When I say 5 prime end, it actually means a 5 prime phosphate. Phosphate group is free here. And 3 prime end, there is an OH group that is free here. So you will always find many people saying 5 prime phosphate or 3 prime OH, 3 OH, 3 prime OH. That is how people talk when you're discussing about DNA. So I will take this one slide, and then I will just have a quick question on, or maybe a few questions we will do. So Chargaff is one remarkable scientist where he, by the time they knew, he's a biochemist, he found out the chemical nature, and there are many more other scientists who isolated the genetic material, or that is the DNA, and then they broken it down into several pieces or its components, and that is how they knew that there are bases, and then there is a sugar, that is deoxyribose, and there are phosphates. Okay, people will still have a trouble about how can a molecule like this function as genetic material or a molecule of information. So something different is Chargaff did something. I don't know why he did, but I think it has been a crucial one for the elucidation of the structure of DNA. What he has found is always there is a correlation. That is, adenine to thymine is almost always 1 or very close to 1. You can see in all of these. The ratio of adenine to guanine, there is not so much of you cannot see consistency. Thymine to cytosine, there is similarly no consistency. But if you take the ratio between adenine to thymine, it always comes to about 1. Guanine to cytosine, there is always a ratio to 1. And again, you see purines to pyrimidines, there is always this ratio. So Chargaff, what he found was the amount of adenine is always equal to that of thymine. The amount of guanine is always equal to that of cytosine. So these are the purines. The amount of purines is always equal to that of pyrimidines. Okay, that is an interesting finding that he has done. We will discuss the structure of DNA in the next class, but I would want you to quickly try to answer these questions in the meantime. I hope you know what is double-stranded DNA. These will be useful too. Double-stranded is like this: 5 prime, 3 prime. This is 5 prime and 3 prime. So we actually have to discuss about the structure of DNA, but I think I'm assuming that you all remember a lot of things from the biology for engineers course. So as a brain teaser, you can assume, you can try to, you know that adenine base pairs with thymine and guanine base pairs with cytosine in double-stranded DNA. If there is A here, there must be a T here. If there is a C here, there must be a G here. So can anybody tell me, in a double-stranded DNA, a DNA was found to contain 33% of G. What is the percent of T?
17%.
17%.
How did you get to that?
G is 33. So obviously the paired with C is also 33. Altogether you'll get 66. And then 100 minus 66 is 34. So 34 by 2 is A and T.
That's right. So you have the answer there. Okay. These kinds of questions will be the norm in all of the exam. As I told you before, you would not be writing essays, not so much. Either you will be doing some calculations like this, or you will be drawing diagrams. So for the benefit, I would suggest you to have your notebook and try to practice along with me. The only reason why I'm drawing the structures is because I hope you will be able to draw as well, and then hopefully you will remember it for a long time. There is this question, it is a little bit tricky, I guess. The genome of an organism has 50% GC content. Pick the correct statement. Anybody?
80 is 50%.
Okay.
Sir, all of the above?
All of the above, actually, because
That's right. All of the above. If GC is 50%, that means this is 25, this is 25. So the remaining is 50%, which should be A. G is 25, T is 25. So all of the above is the correct answer. And you also have to remember that I encourage people to write competitive exams like CSAR and GATE. And for that, this kind of training is very useful. Not only in this course, but in the following courses too. You can always think and think in those slides, how can I frame a question and so on. And hopefully you'll be able to do, you'll become much more brighter and careful about it. Say if we didn't follow the options properly, then we choose the first one that is there, then we might be missing out on marks. Both these exams that I know, GATE and CSAR, they have negative markings. It's not just you knowing. You should be knowing it confidently. That's what negative marks will do, right? Your confidence in the knowledge that you have is what negative marks will prove. If you're making too many errors, that means what you have information, the confidence level is low, the accuracy of the information. Okay, can I have again? This is the last question, I think. I will, yeah, this is the last question. One of the equations is false regarding the composition of bases in a double-stranded DNA.
the third one.
Option C.
Why is that?
A plus G should be equal. Should be equal to T plus C. So we know that that equation is right.
Yes.
So
if I take
can't
this one is 10, and then T is 10, and say this is 20, then this would be 20, right? I'm just giving it for if somebody has trouble understanding it. On both sides, you have 30 equal to 30. So just assuming this one, this is 20 by 20 is equal to 1. So this is also fine. This is a bit tricky. Now, 10 plus, I said T is 10, and C is 20, and G is 20. This is not. 20 is not equal to 14. A, I said 10. G is, I said 20. T is 10, and G is 20. So 30 by 30 equal to 1. So that is correct. So C is the wrong option there, or the false. So when you're reading the question, you should read about this part of the question and the options very closely. What you're seeing right now are actually kind of simple questions, and if you're supposed to do these kinds of questions at a higher rate or speed, then the errors will also increase. The efficiency is what all matters. Some people might take to answer something like this, they might take 30 seconds, somebody might take 3 minutes. If somebody they might take 3 hours because they might have to learn the whole of molecular biology and then figure out the answer, right? So efficiency is what you're trying to increase, which is a factor of speed and accuracy. And typically, speed is kind of inversely proportional to accuracy. If you try to do it very fast, your accuracy may go down, right? And trying to improve both is what is important. So in GATE or CSR exams, you will have, say, something like, I think, I don't remember exactly, you will have 1 or 2 minutes per question. And they may have, their syllabus is vast, and that means you have to have a lot of knowledge about everything and then be very fast. Okay. So are there any questions in any of these? We will discuss that later. Tomorrow, we will in the next class, we will talk about the structure of DNA. Okay. Any questions?